Chapter 3 will look at cell structures of the eukaryotic and the prokaryotic cells with an emphasis in prokaryotic cells. Eukaryotic cells are complex cells with a membrane-bound nucleus and membrane-bound organelles. On the other hand, prokaryotic cells have no membrane-bound nucleus or membrane-bound organelles. Eukaryotic cells are larger than prokaryotic cells. Red blood cells here and bacterial prokaryotic cells here underscore the size difference between eukaryotic cells and prokaryotic cells. This chapter will look at the microbial cells and applications of microbial cell structures as target for antimicrobial drugs with selective toxicity. Understanding the structure and function of microbial cells allows us to control and manipulate the growth of microbes. It allows us to grow them in the laboratory or to create antimicrobial drugs that can harm the microbial cell without harming the host. These drugs are said to have selective toxicity. A drug with a good selective toxicity has the ability to cause harm to the infectious cell without causing harm to the host cells. Selectively toxic antimicrobial drugs target unique and essential microbe structures, causing minimum harm to the human host cells, which are eukaryotic cells. As we study the structure of eukaryotic and prokaryotic cells, consider them under the light of being targets for antimicrobial drugs. Again, a good target for an antimicrobial drug will have to be a unique structure that is essential to the cell. We will start by looking at the outer layers or the coverings that we find in prokaryotic and eukaryotic cells. For starters, all cells have a cell membrane. What are cell membranes made of? You should know by now the answer to that question from Chapter 2. In all types of cells, cell membranes are made of phospholipids. Some cells also have a cell wall superficial to the cell membrane. Is the cell wall a structure found in all types of cells? You should know by now the answer to that question from Chapter 1. Which type of eukaryotic organisms have cells with cell walls? Plants, fungi, algae, protozoans, animals. Which of these groups of eukaryotic organisms have cells with a cell wall? Plants, fungi, and algae are eukaryotic cells that have a cell wall. On the other hand, animals and protozoans have cells without a cell wall. What about the prokaryotic cells? Do cells in the domain bacteria and the domain archaea have a cell wall? By now you know that cell walls are found in both members of the domain bacteria and members of the domain archaea, with exceptions. Remember, mycoplasma species of bacteria which lack a cell wall. Are all cell walls made of the same components? The domain bacteria have cell walls made mostly of peptidoglycan. In the domain archaea, the cell walls are made of various components, proteins, and polysaccharides. Algae and plants have cell walls made of cellulose. Fungi have chitin cell walls. Chitin is a polysaccharide. You should now be able to answer the following questions. Would the cell membrane be a good target for antimicrobial drugs? Remember, a good target for an antimicrobial drug would have to be a unique and essential structure to the microorganism. Are cell walls unique to microorganisms? And are they essential to microorganisms? Polymyxin B is an antibiotic that targets the cell membrane of mostly gram-negative bacteria. However, it is only used as a topical skin ointment. Why can this antibiotic be used internally? Could hypertonic or hypotonic environments be used to control the growth of microorganisms? To answer this question, you would need to review what is a hypertonic or a hypotonic environment and what is the effect of either on a living cell and its membrane. Would the cell wall be a good target for antimicrobial drugs? Again, consider whether cell walls are unique structures to microorganisms and essential structures to microorganisms. Would an antibiotic that targets the cell wall of bacteria be effective against Mycoplasma species? Would an antibiotic that stops peptidoglycan synthesis harm human cells? Next, we're going to consider another outer layer or covering found in some microbial cells. This is the capsule. Some bacteria have a third layer superficial to the cell wall. This layer is made of either protein or complex polysaccharides. This layer is called a capsule. Capsules protect bacteria from phagocytosis by human immune system cells. Other prokaryotic organisms have slime layers. Slime layers are diffuse, poorly defined, easily removed layers of a sticky complex polysaccharide. Slime layers help cells attach to surfaces. Some bacterial cells have short protein filaments that protrude outward from the surface of the cell. These structures are called pili. These filaments help the cell attach to surfaces or to a host cell. Some pila is called conjugation pilas because it allowed cells to contact each other briefly to exchange genetic material. Finally, some macroorganisms are motile. In bacteria, motility is due to the presence of long protein filaments called flagella. The protein that makes flagella in bacteria is called flagellin. Flagella come in different arrangements. Some bacterial cells have one single flagella at one end of the cell. Others have a tuft of flagella on either side, yet others have one single flagella on either side. Other organisms have flagella surrounding the entire cell. Endoflagella or axial filaments are unique to spirochetes. Not all bacteria are motile. Those who are move by means of rotating flagella. Motility in eukaryotic microorganisms can be due to flagella, cilia, or pseudopods. Cilia and flagella are protein structures made of a protein called microtubules. Are flagella antigenic? The flagella of eukaryotes is made of microtubules instead of flagellae, and it whips when it moves instead of rotating. The following is a list of the external structures mentioned. Cell membranes, cell walls, capsules and slime layers found in prokaryotes only, pili found in prokaryotes, motility structures. As you look through this list, consider the definition of good selective toxicity. Drugs must target structures that are essential and unique to the microbial cell. Which of these structures will make good targets for antibacterial drugs? Which of these structures will make good targets for antifungal drugs?